And now we move on to part B, part two, if you will, of compartmentation, where we look at compartmentation manifested in what we call tissues. There are many ways to define tissues. You can define tissues structurally, you can define tissues locationally, and in this case, we're going to define tissues functionally. That is to say, this four schema, if you will, the four major functional groups of tissue. So we're going to break out what makes each one unique, what makes each one different, and the key aspects of these tissues and how they're connected and how they work together. Four primary tissue types include epithelial, very, very important tissue with a lot of diversity in it. Connective tissue, which is defined a lot by what we call extracellular matrix, certainly contractile tissue in the form of muscle. And then lastly, neural tissue, which includes the signaling neurons, but also includes a population of what we call glial cells that also comprise that tissue. In order for cells to collaborate and form appropriate structures relevant to function, you not only have to have the right kinds of cells, but they have to be joined correctly. And that's where the role of specialized proteins called junctions and CAMs. A CAM is an abbreviation for cell adhesion molecule. And so we're going to talk a little bit about a few of these guys that are critically important for proper structure and communication. Did you know that a normal human tissue cell, its default pathway is actually to die? If you were to take a biopsy of some tissue, stick it down in the muscle and take a few muscle cells out of there, those cells, no matter how much nutrients and temperature and humidity you give them, will just die because they sense that they are not in their appropriate locations. That's one of the things that makes cancer so interesting, is that cancer cells lose that default pathway to death. There are a number of genes involved, but some of those genes involve these cell adhesion molecules. So let's go through some representative ones and a few, because we're going to see these in tissue, like particularly in cardiac tissue. We have what's called a communicating junction, or better known as a gap junction. And this is basically a double channel. What I mean by double channel is that most channels in our plasma membranes are a single pass across a single membrane. In the case of the gap junction, you have a protein complex that creates a channel between two plasma membranes. So you can create communication through messenger molecules like calcium. So calcium can diffuse through two plasma membranes and then cause effects in the downstream cell. We have another very important junction. Actually, it's a family of junctions, but this is we're just going to call it one. These are called occludens junctions in some cases, occluding, but our term is tight. And it's tight because what it does is it regulates very carefully certain types of the passage, and we call this paracellular movement. Paracellular. When we get to chapter 5, we'll talk more about this. Paracellular is basically moving from one compartment to another, but not passing through a cell. It passes around the cell. So imagine you have a brick wall, and you have the mortar and the bricks, and you drill a hole through the mortar. Now, whatever is passing through that hole is passing, if the bricks were cells, whatever's passing through that hole is passing paracellularly. So a big time for water, certainly ions and proteins. In the disease called cholera, which often occurs in disaster zones, what happens is cholera causes inflammation in the large intestine, and it abrogates the tight junctions, and so people literally get biblical, and I'm not exaggerating here, biblical levels of diarrhea, and they die rapidly of dehydration. So that's a tight junction for you. And then the third one is actually, there's several here. We have cadherins and integrins, and I'll explain the difference in a second. These guys are involved in anchoring to either other cells, or in epithelial tissue, we call that basement membrane. And again, I'll get to that when we get to epithelial. Cadherins, again, a cell adhesion type molecule that also serves a communication role. It tells the cell it's in the right place in certain situations. But the big one for communication and anchoring is known as integrins. And integrins are often found on the basolateral side of a cell, along the basement membrane, and they serve a very, very important communication role in telling the cell that it's anchored in the right place. So cadherins and integrins are examples of anchoring and or some cell communication. That's why we don't use that word anymore, because anchoring junctions do serve a communication role. And so here we have gap junctions, and they're made up of these complexes of a protein called connexin, and because it's a communicating, and it's a double channel. So we can move across two plasma membranes rather than just one. And where we see this in spades is in certainly smooth muscle, but also the heart. What we see across skeletal and cardiac and smooth is almost like a spectrum. Is that the heart has some aspects that are like smooth muscle, and it has aspects that are like striated or skeletal muscle. So here are some gap junctions. Again, why? Because we want that heart to contract as a single unit. So that's why we have those gap junctions there. And here's an example of a tight junction. You can see the proteins are arrayed kind of like a rock climber's harness. Again, it's designed to be very, very tight. These different colored proteins up here are part of that tight junction, and you actually see different arrays of proteins depending on the kind of tight junction that you have. Turns out we also have some, so there's our tight junction. We also have some anchoring junctions there. These are cadherins and anchoring junctions that we talked about previously. So we see both in this picture. Again, anchoring junctions are more than anchoring. They do have signaling capacity, and there is a dynamic interaction here that it's hard to describe, especially since this is maybe your first time seeing these things. Tissues are often defined, and especially with connective tissue, by their extracellular matrix. An example of extracellular matrix in epithelial tissue is the basal lamina, also known as a basement membrane. Wherever tissue you find where there is extracellular matrix is that the cells are synthesizing and secreting them. Like for instance in bone, you have osteoblasts that are synthesizing and creating new bone. And you cycle through a whole skeleton in like 10 years. A lot of these extracellular proteins have carbohydrates attached to them. So you have glycoproteins, which means you have proteins that have been modified with carbohydrates, and you have proteoglycans, which means that there's more carbohydrate than protein. So the proteins are stuck on the carbohydrate, and the carbohydrates are stuck on the protein with glycoproteins. Lots of what we call insoluble protein fibers. And to illustrate what I mean by insoluble, if you took a piece of meat, for instance, let's say you take a piece of chicken or a piece of beef, and you just dropped it into a glass of pure water. Yeah, you would see that thing get kind of funky after a while. Certainly the water is going to affect the osmolarity and that sort of a thing, but you wouldn't see it just dissolve away. On the other hand, if you took an egg and you separated the egg white out from the yolk and you drop that albumin into water, you would see it diffuse and it would diffuse out and eventually become part of the water solution. That's what we mean by insoluble, is that they will not dissolve in an aqueous solution. So things like collagen, which you've probably heard of, fibronectin and laminin. These guys are for strength and structure oftentimes. They do have some communication role, but by and large the structure is usually the most important point when it comes to these insoluble protein fibers. That said, things like collagen and laminin can induce clotting if they're introduced into blood plasma. So they do serve a communication role. Also, extracellular matrix can be mineral, like in the case of bone, or even ceramic type mineral with teeth. This is an example of actually uh, cartilage. And so we see quite a lot of space in between these cells because there's a lot of extracellular matrix. Bone, you can see the cells are very, very organized. And again, we have that mineral outside. It's hard to underemphasize the importance of good extracellular matrix. This is just along the same lines as the cells themselves. These cells need to know that they're in the right place. It helps the cells maintain their integrity and so forth. And here's an example. If we have healthy myocardium, there is a robust extracellular matrix full of different kinds of proteoglycans and glycoproteins. But if we have a myocardial infarction, not only are the cells potentially impacted, and even if the cells don't die, they may lose function because of the destruction and altering of the extracellular matrix. A big theory as to why heart can't seem to recover from severe ischemia, like in the case of an infarction, is not only the damage to the cells, but the damage to the extracellular matrix. So there's a lot of research in to see if we can't somehow replenish that extracellular matrix so the damage is less severe. Our first major category now, moving on into the distinct types, is epithelial. And epithelia, epi means above. If you've heard of epidermis, it's the outer dermis or the higher dermis. Epithelial tissue is almost always on the outer surface of organs or the outer surface of the body, outer surface of tissues. It's one or more layers. If we have a simple epithelial tissue, it means it's one layer. If we have stratified, or as they call it, it's a pseudo-stratified, I'm not going to go there today. If we have more than one cell layer, then we have the stratified. And so it can be one or more layers, and those number of layers is directly related to function. Epithelial tissue always has, or almost always has, a basal lamina, a basal membrane, if you will, of extracellular matrix. And again, that's there to help hold the tissue in place. It's there to tell these epithelial cells that they're in the right place. There are a number of ways you can sort of examine this on your own. If you've ever, like for instance, if you have chicken wings or something and there's a layer of skin or that sort of a thing, if you peel that skin off, you can actually see that sort of basement membrane that's kind of holding that tissue on there. Epithelial tissue can be sheet-like, where it's serving as a cover, or and again, this is an arbitrary thing. This is humans imposing on the spectrum of function, if you will. They've chosen to include secretory tissue as part of epithelia. That's because secretory tissue is in fact found often associated with these outer tissue layers. If you look at your skin, you would think of sebaceous glands and sweat glands, or if you look at ductal epithelia, like for instance in your pancreas, next to the ductal epithelia are secretory types of cells. And so it's part of the epithelial spectrum, either sheets and or secretory function in these cells. And here's an example of a sheet-like surface. Okay, this is a squamous epithelium. This is found inside your lungs. And here's some secretory epithelia. In this case, it's a ductal secretion, an exocrine gland that secretes into the center duct and that transports whatever is being secreted into digestive tract or perhaps the outer surface of the skin. So here's a simple epithelium, a single cell layer. And here you can see this sort of slightly reddish area. That's our basement membrane, basal lamina, extracellular matrix, cell adhesion molecules. The cell adhesion molecules down here are often different than the ones we would see up here. So most epithelia attach to a matrix to an underlying basal lamina, and that helps hold it in place on whatever organ or tissue that it's located. We classify epithelia by its structure, how much it's layered. So I said simple or stratified. And again, there are some in-betweens. It's an artificial construct trying to label these sometimes. The shape. So for instance, the squamous epithelia looks kind of irregular, but somewhat rhomboidal occasionally. You can see square cube shapes or these long cube shapes that we call columns. So we often say simple squamous or stratified squamous, et cetera, et cetera. So here are just some examples of that, simple cuboidal or stratified squamous and so forth and so forth. Again, these one layers or multiple layers and the cell shapes often are directly related to the function that they engage in. So here's some examples just to figure for you. So one of the key questions when it comes to epithelial tissues is, what do these tissues do? And over time we have developed about 5 major functions: exchange, transporting, ciliated, protective, and secretory or secretory. Exchange, what we're trying to do is facilitate rapid diffusional movement across that epithelial layer. You can see it's very, very thin, very often used for gas exchange, or in some cases fluid exchange. And we'll talk about that a little bit later in the class. Transporting epithelia are like exchange, only instead of facilitating rapid diffusional events, they often involve what we call facilitated diffusion. That is to say, they're using proteins. In some cases, they're doing active transport. It doesn't have to be through the cell. It can be either through the cell, like in what we call transcellular transport, or if you remember the tight junctions, it can be between the cells. So tight junctions can regulate movement between the cells, and that's paracellular transport. We also have ciliated epithelium, which is like transporting, but instead of being vertical, it's transporting it sideways. So we're moving things laterally. Examples of ciliated epithelia include the fallopian tubes, transporting eggs from the ovaries to the uterus, but also the airways. Have you ever heard a smoker go, huh, huh? They're always sort of huffing. Not huffing material, but they're always kind of, doing that. That's because the nicotine and the tar and the smoke particles contribute to, first of all, a paralysis of the cilia, or in some cases outright death of the ciliated epithelium. So they can't move that material as well, and so they have to use expectoration type reflexes to get that material up to the epiglottis. But we're not really moving vertically, and this arrow is going to go fast and go away. Sorry about that. We're not really doing a lot of, that would be more of a transporting situation up here. And don't be confused by the cilia here versus the wavy line here. Those are villi. Those are different structures, and villi are meant to actually increase surface area. Cilia, on the other hand, are generally microtubule structures that move laterally in a wave-like motion. We have protective epithelium, and then that key characteristic there is the multiple cell layers. And so more cell layers means more durability, more waterproofing, more ability to absorb strain. And then finally we have the secretory or secretory. This happens to be an exocrine one, and I'll show an example of an endocrine a little bit later. This is just a picture from your book showing you examples, and I'm going to show you the larger versions of these here in a second. But you have this beautiful, if abstract, representation of all the different epithelia. Exchange epithelia here in red. This is endocrine secretory epithelia, and this is exocrine secretory epithelia. Ciliated or perhaps transporting. I think the purple is transporting in this. Ciliated would be the fallopian tube, so that's shown in light blue here. So transporting would be these purple ones, and that's because of the movement of materials either from compartment to compartment in the digestive tract. So here's our exchange epithelia. These are endothelial cells in capillaries or inside all of our blood vessels, interior surface of the heart. In the epithelium in capillaries is very thin and meant to facilitate rapid diffusion of gases and nutrients. Exchange epithelium. And here are a couple of pictures. So endothelial cells, the squamous cells inside the lungs are also exchange epithelium. Here's protective epithelia for you. This is actually tissue. It's more than just the epithelium. So inside that box is the epithelial component of this organ system. Multiple layers of material. The more layers often mean more shear, more stress and trauma on that tissue, and it adapts to that strain by adding more layers, which is why you get things like calluses. Ciliated epithelium, again, it's for lateral movement, like fallopian tubes, like I said, or in the airways are the two main ones that we talk about. Here is secretory. This happens to be exocrine in nature. Endocrine tissue, I'll show you on the next slide. Here's endocrine tissue, this lighter area. This is known as an islet of Langerhans. This is full of about, I think it's about half a dozen different types of cells, maybe 6 or 7 different types, a bunch of different hormones that are secreted directly into the bloodstream. So we actually have some capillaries in here that you can see. Those darker areas are actually small capillaries. And what it's doing is it's secreting it out of the cells into the extracellular fluid and by extension into the blood plasma. Over here, the darker purple, this is exocrine epithelial tissue. These cells secrete bicarbonate and digestive enzymes into these ducts, and you can even see the ducts in between the cells, into this larger pancreatic duct going down to the duodenum of the small intestine. So we have exocrine and endocrine in the same picture. And here's an example of transporting epithelium. Again, you see that villi surface. This is the luminal surface. We often will use that sort of squiggly line up there to illustrate the luminal. The other word is apical. So the apical membrane is the part of the plasma membrane that is on the luminal side of an epithelial layer. The other little giveaway is that the tight junctions are often right next to that apical layer. So here we have the apical layer, we have our tight junctions, and then this membrane right here can be very, very different. If you remember, I talked about polarized epithelia in an earlier lecture. This is the basolateral surface of the epithelia. So if we are on the interior surface of the tight junction, we call that basolateral. And if we're on the luminal or the exterior region versus the tight junction, that is the apical surface. Moving on to connective tissue. A lot of connective tissue is involved in structure and strength or impact absorption. The main variability in connective tissue, and again I'm generalizing a little bit here, is the extracellular matrix, sometimes referred to as ground substance. That's a little bit of an older term, but extracellular matrix is very much variable. The proteins have sponginess or they have stretchability. Some extracellular matrix is good for tension, like for instance your tendons and your ligaments. On the other hand, like for instance cartilage is very good for compression. The cells are generally fixed, though there are exceptions, like for instance blood is often lumped into connective tissue. Those cells aren't fixed at all. But if they are fixed, we often refer to them as things like blasts if they're making more of that extracellular matrix, or clasts if they're breaking down extracellular matrix, and or just generalized cells we call cytes that are neither depositing nor absorbing extracellular material. Cells can be mobile, so we don't usually call things blasts and so forth. If they're mobile, often refer to them just as cytes. The insoluble proteins we often see, collagen, elastin, fibrillin, fibronectin, fibrin is in there as well. And here are just some examples for you. There's cartilage, there's bone. We've seen these before. Here's blood. Again, a very watery extracellular matrix. A very, very watery extracellular matrix, whereas bone is almost the exact opposite. It's crystalline, ceramic in your teeth. This one's kind of a fun one. This looks like skeletal muscle to me, but it's actually some tendon or perhaps ligament. I forget now. It's been a few years since I put this in, but you can see the regimental extracellular matrix. So we can see a lot of variability across these samples in these pictures. And I've got these here. I'm not going to, you can refer to these on your own for additional understanding. Here's blood. Again, we not only have red blood cells, but we have white blood cells and different kinds and lymphocytes. Additional connective tissues. These are an interesting family. Adipose connective tissue, unlike the other connective tissues, doesn't have extracellular matrix. The matrix, and I don't even want to call it matrix, what they have is large amounts of material found inside the cells. And they do serve a connective purpose. I had a close friend who passed away, unfortunately, from complications secondary to anorexia nervosa. She lost so much weight that she lost a lot of the fat that cushioned the outside of her heart. And it actually created a rub point between her heart and her aorta, and she unfortunately died of that condition. So adipose does serve a very, very important connective role. It just doesn't have that extracellular matrix. We have two major kinds. We have the white fat. That's the fat you're used to talking about. That's the fat that nobody really likes all that much. But I just illustrated to you that it has life or death consequences. Somebody who has untreated anorexia nervosa or severe bulimia or a nutritional deficit in the case of starvation type syndromes, loss of white fatty tissue can lead to all sorts of complications. Brown fat, on the other hand, is really, really interesting. Brown fat is actually thermogenic in nature. So if you decided to do an internship down in Antarctica, or you decided to move somewhere like Norway, what would happen is the small areas of brown, you find this down subscapularly underneath your scapular bones, will actually grow. You'll actually gain fat while being in a cold thing. And I have some theories about this, that because we live in warmer temperatures, we're not tolerating the cold as much, and we don't have as much brown fat in order to generate heat. The brown fat has an uncoupled respiratory pathway. It will go through the citric acid cycle, but it won't go through electron transport nearly as much. And so it'll generate a lot of heat, relatively little amount of ATP. So it's a thermogenic tissue. But again, you don't see extracellular matrix, even though this looks kind of weird. Those are just multiple droplets inside the cells. Blood is also an additional connective tissue. We've already talked about that. Given that we're going to spend a fair amount of time on muscle tissues later on, I'm just going to go over the highlights here. Muscle tissue has the property of being both excitable in terms of the voltage across the membrane. We'll talk about that as well as being contractile. That is the ability to impart force and movement on other cells and tissues. The three main types we have are skeletal, that's the most by mass, smooth, and cardiac. So cardiac, what we have is you can actually see it quite well here, is that you have mononuclear cells, and these little lines that you see, those are called intercalated discs, and that's where you see those gap junctions. Here's one with an arrow next to it. That's where you have a connection between these two cells, and they literally share cytosol. They share, in particular, calcium ions flow from one side to the of these junctions to the other. As opposed to smooth muscle, and this one's kind of hard to tell. Smooth muscle is also mononuclear. One thing that's tough to see in the cardiac is that the cardiac is actually striated, and that's the difference between a striated muscle and a smooth muscle. Is that smooth muscle, the actin and myosin are not arranged in regimental order, like you can especially see here in skeletal muscle. So skeletal muscles form. They fuse together. The cells, the precursor cells, will fuse together, and you get these polynuclear cells that are much, much larger than your typical human cell. So they have multiple nuclei as opposed to uninuclear in smooth and cardiac. Both cardiac and skeletal are striated, even though it's hard to see in this picture. So you can see there are sort of similarities between cardiac and skeletal, and then there are similarities between cardiac and smooth. There's sort of a gradient here. Nervous tissue or neural tissue, we have the excitable cells, neurons, involved in signaling. And we also have glial cells, which are involved in metabolic and structural support. So here we have this very, very large, this is a somatic motor neuron. Not all neurons are this big relative to other tissue, with its big axons going out of here, sending signals to other cells, whether it's other neurons or to muscle tissue or to glandular tissue. And then we have the glial cells surrounding and supporting these neurons. Glial cells are not directly excitable, but they can modulate excitability in a neuron. They work very, very closely with neurons. And the more we understand about glial cells, the more we appreciate their role in neural function. Here's a lovely Purkinje cell. Purkinje cells are known for their lots and lots of different processes because what neurons are doing are integrating. That is to say, they're integrating a signal that's coming into the central nervous system with the proper effector and response systems in other tissues. Now you have this lovely chart, or you may have this lovely chart. I haven't looked at the latest edition. I would just say warning on this, and you could sit here and memorize this stuff all day, but I don't think that's going to do you a whole lot of good in this class and in the big picture of things. There may be a time when you will have to memorize some of this if you take a histology class or something a little more specialized, but for our purposes, a chart like this in the book is merely just a reference point. If you want to double check something, make sure you're on track with something, this makes a good one to double check, but it's not really worth memorizing. The half-hour point right now, and I have about 11 slides to go, probably some more than others. I just want to touch on a few of these subjects and cover the highlights, and then hopefully we can wrap up here pretty soon. Cell growth and division is important, but equally important is cell death. If we have too many cells, that's what we call hyperplasia. And if those cells are dysregulated, we call that dysplasia. So cell death is as important to life in many ways as cell growth and division. We have two major kinds, and trust me, there is a spectrum between both of these. So these are the ends of the spectrum. We have what is known as necrosis, which is caused by either disruption of the cell in the form of trauma or injury, like poking or some other stress injury of some kind, or you can interfere with the metabolic pathways of the cell. We call that poisoning collectively, where some sort of molecule is interfering with the normal metabolic function of the cell. We also have at the opposite end of the spectrum what is known as apoptosis. Cell suicide is a poor choice of words, I think. What we mean is intentional cell death, programmed cell death. These cells die because either their default pathway to die is activated or some other signal is inducing them to undergo cell death. Here is a little bit of a set of differences. Some things I would point out to you is that apoptosis is actually quite fast and it is not inflammatory. That's among some of the key things. It's actually pretty fast. It'll happen in a few minutes or an hour, whereas necrosis tends to be slow. It is very inflammatory and it will cause inflammation in surrounding tissues. And it is basically characterized as dysregulated cell death. What seems to happen is that you lose the ability to make ATP, calcium can't get regulated, and it just causes a dysregulation of the metabolism in the cell that's inflammatory and relatively slow. Apoptosis, on the other hand, is highly regulated and non-inflammatory. It uses ATP, and so the mitochondria actually work right up until the very, very end. The cell it blebs, it creates these balls of membrane that are eventually phagocytosed by other cells, and it's not inflammatory. Some roles for apoptosis are very, very important in bone shaping. So if you take 305 or an osteology course, you're going to see all of these uniquely shaped bones, and you'll probably spend a fair amount of time identifying and differentiating them. Well, apoptosis is a big factor in that. Occasionally, very rarely, you'll see a person, and sometimes they'll have corrective surgery for this, take your hand and just put it in front of the screen and then spread your fingers out. If you look in the right middle picture there are people that continue to have webbed digits because for whatever reason the apoptosis of the tissue between your fingers didn't activate, and so they were left with that residual tissue that normally dies off by non-inflammatory death. When a frog or a tadpole turns into a frog, it absorbs its tail. That's because of apoptosis. If somebody gets, if a female gets pregnant, what happens is the mammary tissue grows and develops, and so the breasts get a little bit larger. And they start producing milk. Well, at the time that the milk production is no longer needed, that tissue that creates the milk actually dies off in a non-inflammatory way, and that's apoptosis. So here are some examples for you. This is just a graphic rendition of what I've already told you. Necrosis is slow, dysregulated, and inflammatory, often involving calcium release. Apoptosis is highly regulated, uses ATP. The cells are basically turn into small balls of membrane that get phagocytosed by other cells. I actually accidentally induced apoptosis in some cells I was photographing under a microscope, and it looked like a firework. You saw the cell condense, and then you saw these blebs going off because I had this fluorescent label inside the cells. It looked like a firework going off. It was really, really cool. I had taken a photo every second, so it looked just like a firework on the 4th of July. A little bit about stem cells, because stem cells are a growing aspect of therapy. Stem cells are cells that have the ability to differentiate into either more of itself or many other different cell types. Stem cells retain replicative ability, whereas highly differentiated cells like, for instance, skeletal muscle or cardiac muscle or neurons lack the ability to divide. In some cases, some cells retain the ability to divide, but they have to de-differentiate first. They may have some ability. For instance, there are some neurons in your nose that have the ability to divide and regrow, but they have to de-differentiate first before they can divide. The key aspect of cancer, by the way, this de-differentiation and this stem cell-like morphology. It's basically they're hard to detect because they just don't look like anything. They're just sort of nondescript cells. The different types of stem cells, a fertilized egg or a cell from that embryo up to around day 4 following conception can form a complete human being. That's what we mean by totipotent, is that these cells retain the ability to divide and differentiate into an intact organism. We get perilously close to having totipotent cells in the female egg. There is a form of tumor called a teratoma, which is about as close to asexual reproduction as you'll ever see, because these tumors will grow hair, they'll grow teeth, they'll grow heart muscle. We had a section of teratoma in a histology lab that I was doing, and we identified 14 different types of tissue in that teratoma. Thankfully, teratomas don't form intact human beings, and they are not necessarily lethal because the cells are really differentiated. Pluripotent means that they can form many different types of tissues. Like, for instance, maybe they can form bone tissue or cartilage or blood cells or that sort of a thing, or fat tissue. So pluripotent means lots of different tissue types, whereas multipotent is capable of differentiating into different cell types within a given tissue. So for instance, try to think of a good one for that. If you think about the islets of Langerhans, some of those cells were multipotent, capable of differentiating. Red blood precursor cells are what we would call multipotent. That is because they make blood cells. It's just different kinds of blood cells, whether red cells or white cells. That's a good example. The hematopoietic cells that create red and white blood cells. Going to wrap up with a few short questions here. We're just about done, running just a little bit long. But look at skin sometime. If you can go get a picture of skin, take what you've learned in this chapter and apply it to skin. What do you see when you look at the cell organization? Do you see muscle or epithelial or neural or connective tissue? Can you spot these things? Can you find extracellular matrix? This is not a histology class. I'd love to be able to do some of that. It's a neat class, but I just want you to think in that direction because you'll probably experience it at some point. And here's a picture for you just to kind of include on that. What types of tissues do you see in here? Can you relate form and function, if you know what I mean? Same thing's true with this slide. I'm not going to belabor it all that much since I'm running a little bit long. In summary, functional compartments. We talked about macro compartments and we talked about fluid and functional compartments. We talked a little bit about membranes and the intracellular compartments. We talked about the four major tissues, the remodeling in terms of apoptosis and necrosis, as well as the types of stem cells, and just wrapped up with a little bit of reflection on organ systems. So thank you for your time. That's chapter 3, and I will hopefully see you soon.